Now here is the link between water and the legal tender of the body, the neural liquid.
The brain contains 100 billion cells, neurons, and they make 100 trillion connections or synapses. And each of the neurons release chemical substances called neurotransmitters. And these neurotransmitters are the legal tender of the brain. That's how these cells communicate with each other.
Memories and visionary thought are created in the cerebral and frontal cortex as the neural liquid of dopamine travels through precious metal transmitters into NMDA receptors on either end of the dendrite neurons to create the holographic images as this neural liquid crosses the synapse we call our mind's eye. The greater the amount of a dopamine, the greater the number of active dendrite neurons and dendrite pathways, thus making us think clearer and more accurately as we consciously see ourselves and the life we are leading. The neural liquid of dopamine can be thought of as a liquid crystal similar to the liquid lighting up an LCD screen. As you can see, the nucleus of each dendrite neuron is DNA, which coordinates the beam of light we internally see through our mind's eye, which passes this movie clip to our hippocampus, which relays this information with all the associated emotions and opinions down our spinal column. Each disc along the spinal column relates to a dendrite pathway leading to a particular part of our body. Along this pathway, our memory is saved and the neural pathway changes from a dendrite pathway to an axon pathway. And the neural liquid, a dopamine, changes to serotonin, which coordinates our body's vascular and lymphatic systems to move our bodies and eliminate waste. To know if this neural liquid is traveling good and strong within you, ask yourself, how well do you dream? And how do you feel when you do dream? The answer you give yourself will always be the same. You want to dream better, for this is an indicator of your own consciousness. Now we can see and understand our bodies are controlled by our neurons, and science is able to produce stem cells to improve human life on this planet. Yet the dilemma is who should receive these stem cells, and do these stem cells solve or just delay the problems of neural miscommunications?
There are a whole series of new ideas that have come out of this. The fact that prions are infectious proteins is totally new. This is unlike all other infectious agents. Prions cause brain degeneration. They cause sporadic, genetic, and infectious forms of these diseases. There is no other disease paradigm in which you have both genetic and infectious diseases. And prions are the most well understood among all the neurodegenerative diseases, which include, of course, Alzheimer's disease and Parkinson's disease, which are many thousands of times more common than the prion diseases. Now, it's the discovery of prions and many other new findings which allows us now, I think, to define neurodegenerative diseases in terms of their cause, not the effect. So I think most people would agree now that degenerative diseases of the nervous system are disorders of aberrant protein processing. So the proteins are being processed abnormally. To give you a little idea of the numbers of cases, so we have 400 cases of prion disease annually in the US, but there are 4 million people with Alzheimer's disease, about 1 million with Parkinson's disease, and about 20,000 with ALS or Lou Gehrig's disease. So these are huge numbers. Now, there are many, many similarities between these diseases. What we see are these abnormal protein deposits. I showed you PRP scrapie in the brain, and I showed you these PRP amyloid plaques. Well, the brains look very similar if we use antibodies, but these are different proteins now, but the structures look very similar in Alzheimer's disease. They're slightly different in Parkinson's disease and ALS. But what's so interesting about all of these is that the mutations in the genes that we find in familial forms of the disease encode the proteins that are found in these protein deposits, except here this is less clear with ALS. So the accumulation of misprocessed proteins causes the nervous system to malfunction, resulting in problems such as dementia, we're back to this word, Alan, difficulty moving, and weakness. And preventing the accumulation of these misprocessing proteins provides, I think, for the first time, a rational approach to treating degenerative diseases of the nervous system. Prions are unlike all other infectious pathogens, viruses, viroids, bacteria, fungi, parasites, all of which multiply by having an RNA or DNA genome direct the synthesis of progeny pathogens. So the precursor of the prion is an alpha helix-rich protein, PRPC, that's synthesized in what's called the ER, or endoplasmic reticulum of the cell. The infectious form of the prion protein, PRP scrapie of the prion, is rich in beta structure, and it is formed from PRPC on the surface of the cell in these cholesterol-rich microdomains. It's a kinetic race between the formation of PRP scrapie and the cell's ability to clear PRP scrapie. And that when the cell can keep up with the formation and clear it, like it does with all other proteins, in fact, when that happens, everything is functioning fine. But when the cell gets out of balance, it can no longer clear PRP scrapie at the rate that it's formed. And we're talking about very, very low levels that we can't detect even by these animal assays. Then something goes awry, and we began to accumulate more and more PRP scrapie, and eventually the animal gets sick and it goes on to die, or the human being. Something happens to the signaling mechanisms in the cells, and whether that happens because PRP scrapie is on the surface or whether it's deep in the cell and it mucks up the signaling mechanisms, I don't know. That's a very simplistic answer to a very complicated question, but I can't answer it.
We can simply say that when our bodies are young, the crystal-like nanowired neural pathways can create an abundance of correctly folding proteins that are engineered to make us feel and look fabulous, as well as making us behave in a light-absorbing and learning behavior. Whereas as we age, the crystal-like nanowired neural pathways start to refract our internal guiding lights a little differently. As these pathways lose their conductance and light-absorbing properties, as outside pathogens enter our bodies through the food and water we consume, these neural pathogens are different and similar than normal pathogens. Normal pathogens operate on a chemical level. We see them and we remove them either surgically or with our immune system. Neuropathogens don't operate on a chemical level, but rather on an energetic level. These particular pathogens are not something that we can see under a microscope and we can physically remove them, but rather they affect the movement of light waves, similar to the way our body's neurons affect every bodily function, protein production, or every mental thought put into action. Science has labeled these pathogens as prions, and every neurological disease can be directly attributed to our body's neurons' inability to defend themselves from the many variety of prions. This leads me to the belief that the thought precedes the motion, even in the smallest of pathogens. To define these prion thought waves, or protein folding waves, we can simply understand these prions have the ability to instruct the light to move in a wave pattern similar, yet different than the wave patterns of our body's neurons.